genetic variations. Variations between individuals of the same species can be due to the differences in the sequence of the nucleotides. The question arises that how genetic variations emerge. There are different ways that the genetic mutations emerge in the sequence of the DNA. This is the first reason that genetic variation can be due to the mutations. Mutation is a phenomena that usually occur in the genomes. Here we can see that the mutation rate is higher in case of mammals. So in case of mammals, the mutation rate is high and the genome of the mammals is also large in size. While if we come towards down side, that then the mutation rate is found in case of invertebrates and plants, then unicellular eukaryotes, then eubacteria and archaea. So mutation is the reason that is responsible for the genetic variations which are found in different individuals of a species. Another reason that how genetic variations occur, the second reason can be the recombinations. That there are recombinations and shuffling of the gene variants which are also called as alleles in the population. So within the populations or within the individuals, different alleles or different gene variants, they shuffle. So that is also one of the reasons of the genetic variants which are found in different genomes. Genetic variations is due to the recombination. Recombination is a process which is usually seen in case of meiosis, where the chromosomes, they change their fragments. Here we can see that the chromosomes in the form of sister chromatids or non-homologous chromosome, if they change their fragments, that is due to the recombinations. That is an event found in while during the cell division. So during the genetic variations, there can be good mutations, there can be bad mutations. So good mutations usually spread throughout the population or throughout the individuals of a population. And other reason of the genetic variations can be due to the gene flow. So here is population number 1, here is population number 2, that is shown in the form of animals. Sometimes certain alleles, they move from one population to another population. Here we can see that 2 allele variants, they move from one population to another population. Same is here in this diagram, that 2 animals, they move from this area of the population to this area. So due to the movement of alleles from one population to another population, which is called as gene flow, is responsible for the genetic variations. The fourth type for the genetic variations can be due to the genetic drift. Genetic drift is actually change in frequency of a gene variant in a population due to a random sampling of the organisms. In the first generation, here we can see that there are 3 alleles. Allele 1 marked with yellow, allele 2 marked with gray, allele 3 is marked with red color. So there were 3 different alleles in generation 1, but at the level of generation number 4, we have seen that the allele which was marked with red color disappeared. And at the level of generation 5, we have seen that the alleles which were present, which were marked with a gray color, they also disappeared. So one of the reasons of the genetic variations due to the genetic drift. Here are the examples of the genetic variation. The best example is of human skin color. We can see that these in the globe, these are the areas where the skin color is white. Same is here in case of North America. But in case of Africa and parts of the Australia, we see that the skin color, they are black. So the black color and the white color is due to the genetic variants or the alleles of those genes which are responsible for the human skin color. Another example of the genetic variation is yellow-brown hair, which are usually found in Australian natives. This part of the Australia has brown or black hair, while this part of the Australia has a light or yellow hair color. So this is due to the alleles which are responsible for the color of the hair. Genetic variations in the form of microsatellites. 377 autosomal microsatellites loci were genotyped in 1056 individuals from 52 populations of 7 regions of the world. These are the region 1, region 2, region 3, 4, 5, 6 and 7. And it has been observed when including all the 7 regions, the variation between the microsatellite markers were 46%, while in case of different regions, the frequency, let's say in case of region 1, it was 7.43, in case of region 2, 9.69, region 3, region 4, region 5, and region 6, respectively and separately. But by including all the data, it has been observed that the variation of these autosomal microsatellite markers were 46%. Genetic variations in case of humans. 2 genomes are roughly or approximately 99.9% similar in case of humans. If we take the example that if human genome is 3 billion base pair, then it means there are 3 million differences between any 2 genomes. So this is the just difference of 0.1%. So due to difference of 0.1%, there would be approximately 3 million differences in the genomes of 2 individuals in case of humans.